Okay, let's give students a minute to log in. Let me go ahead and pause that. Let's start recording and begin. Share my screen. So if we look on the syllabus, why do I have two of those there? We're doing today Lab 1, the ubiquity of microorganisms. Also note that you must finish the online plagiarism quiz by 11:59 p.m. this Saturday, January 9th. You must achieve 100% on the plagiarism quiz, but you can take it multiple times, at least 5 times. If you don't obtain 100% on the plagiarism quiz, then you will get 0 points on the infectious disease project. Okay? Also, you should read about the infectious disease research project in the file called Project Instructions on Canvas for next week, because I'm going to talk about the infectious disease project next week. Let me go there and show you. I think it's under Week 2 Lab. There it is, the Infectious Disease Research Project. You should read that material. This is a 100-point assignment, and we'll talk about it next week, and you'll be starting next week on that project. And it's due, I think, the 11th. February 20th. All right, any questions about any of that? What we're doing? If not, I'm going to shut that. Let me see what this is. Oh, Lab Module 1. Okay, good. This is the lab we'll be talking about, the ubiquity of microorganisms. You can click on the link to this and take a look there. Aha! Bringing your dog to the lab. So you do need to pre-read Torah. If you don't have the lecture textbook, I got a note from a student saying that the bookstore was all out. You can make attempts to get the textbook, but I have copies of the first chapters online. Let me see if I can find where that is. I think it's up at the very beginning. Yeah, textbook chapters. Click on that link, and it's under general course information. I actually wanted the library to have that in the course reserves, and they don't want to do that, so they gave the information to me. All right, let's move back. So you need to do some reading in the textbook, and there's some supplemental material of video clips throughout the lab module. Primary learning objectives. Know that microorganisms are almost everywhere. Describe the purpose of culture media. Describe the terms colony abundance, diversity, and morphology. Define the following terms: inoculation, ubiquity of microorganisms, what's something that's sterile, we're talking about like media, sterile media, what is isolation, incubation, in vitro, like in vitro culture, what's a culture media, and there's different types. There's a broth, there's a media with agar in it, there's a plate, so you have a culture media on a plate, you can have a slant and a deep. And I don't know if we talk about that today, but those are two different types of tubes. There are fastidious organisms, and then I guess non-fastidious organisms. There's non-selective media, which we generally call general purpose media. what's ubiquitous, and what's a colony. Any questions about any of that? So, where can I put this best? Shrink that down a little. So with microorganisms, you should know that these are organisms that are too small to be seen with the naked eye. You need magnification in order to see them. Viruses and prions fall under microorganisms, even though they're not organisms, because they don't have cells and they're not, well, some people say they're not alive. Some people, we're not going to get into that argument. Helminths also fall under a microorganism, and by that we mean worms, and the infective stage of worms are microscopic, as well as the early stages of worms. And microorganisms study worms, helminths, because they cause human disease. Any question about any of that? You should know the phrase ubiquity of microorganism refers to the fact that microorganisms can be found almost everywhere. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn about some basic lab protocols, including using solid media, how to label cultures, procedures for disinfecting the lab benches, and procedures for washing your hands. Here's a question for you. When you're washing your hands to be absolutely clean, because you're not going to be sterile, but to be clean, how long should you wash your hands with the soap? Anyone know?
30 seconds.
30 seconds.
Okay. Culture medium, also known as a growth medium, when it's given to you, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms. How we make this sterile in the lab is before we would give it to you, it would be put in the autoclave, killing all living things inside of it. So it'd be sterile. A culture medium may be a liquid, in which case they're called a broth, and that'll be a liquid culture medium or growth medium. They're commonly used in test tubes. It's not good keeping a broth in a petri dish because it just doesn't work well. It'd be too easily to get a spill and get contamination. But in a tube, it works well. You can add agar to a liquid media to result in a solid medium. And so we also have our growth mediums, culture mediums, sometimes solid. And usually it's because we've added agar to it to make it a solid. So you're going to be watching a video on preparing pour plates. Watch that video. And there's a few questions for you to answer there. That's just not turning in, but just for you to answer. You should know that today we're going to be talking about general purpose media. They are non-selective media that non-selective in function, meaning they don't select for certain organisms or things to grow in them. They're usually complex, meaning we don't know their entire composition, and they support the growth of many organisms that we call non-fastidious. A fastidious organism is one that's difficult to grow, and you need to add certain ingredients to the media in order to grow a fastidious organism. Okay? A general purpose media often simply have peptone, we'll talk a little bit more about that later, and yeast and beef extracts in them to give the nutrients for the microorganism to grow. Any question about any of that? 3 examples of general purpose media include nutrient agar with yeast extract added, and we usually abbreviate that as NAYE, N-A-Y-E, tryptic soy agar or TSA, and then nutrient agar, or NA. Those are three common general purpose media. And they're cheap, and they allow many things to grow. Not everything, but they allow many things to grow. And they're good just because they're cheap. When you use something in the lab, you should always make sure that it is appropriately labeled. That includes petri dishes and petri tubes or tubes. And to correctly label something, you must include your name or your initials, the date, the type of medium, like you could write NA for nutrient agar with yeast extract. The incubation temperature is optional, and you can include that. And you should put what the inoculum is. If you know what it is, like E. coli, just write E. coli. If you don't know what it is, just write what the inoculum is, like air exposure. You expose the plate to the air, and then you grew things from the air. Any question about that? For labeling a plate, you just label on the bottom of the plate, around the edge, like this. Trying to show you, this is written by Travis, E. coli. The date there, I can't read the year, but it might be 2001. And then the media, and then the temperature it's grown out. This is optional, 37 degrees Celsius. For a test tube, you label on a piece of tape and then put tape around the test tube. Any question about that? The trouble with tape is you have to remove it when you're going to put the tape to be discarded. Inoculation is the placement or the transfer of microbial specimens into or on a sterile culture media to get them to grow. There are a number of ways you can inoculate, and we'll talk a little bit about that in this lab. Incubation is just the placement of the culture media in an appropriate place to grow the culture. Usually in the lab, now obviously you're going to be doing this online, but usually in the lab we put things to grow in an incubator. Okay? The incubator usually warms things up and gives it a little bit of moisture. You can have much fancier incubators giving specific oxygen and carbon dioxide percentages and specific temperature. Usually not other things, but humidity, they do that. All right, let's move down here. Some notes. When you're incubating an agar plate, if you were to incubate it right side up, let me show you something. So this isn't a plate, but I don't have one here because I'm obviously at home. And so you can see this. Let's pretend this is a petri dish, and this is right side up. If you were to incubate the plate in the incubator right side up, from the bottom, which has the agar, and that's where the moisture is, the temperature would bring the water out, which would then condense on the lid of the plate, and then the droplets would form, and then they would fall back on your culture, dispersing and potentially ruining your culture. So you don't want to grow agar plates in the incubator right side up. You want to put them in the incubator upside down. And now the agar is on the top of the plate. And so if moisture were to come out of the agar, it's going to condense on the top of the agar, and droplets won't form because the agar will wick the moisture throughout the agar. And even if droplets were to form and to drop on the top of the plate, who cares? We're not growing any bacteria on the top of the plate. So when you incubate plates in the incubator, they should be incubated upside down to prevent moisture from forming on the lid and then dropping into your culture. Okay, any question about that? You should also note that bacteria colonies tend to look different from fungal colonies. Bacteria colonies typically are smooth textured surfaces. There are some exceptions, but most of them are smooth, textured, and smaller than fungal colonies. Fungal colonies tend to be furry or hairy looking, and they tend to be larger also. Please realize that a colony is made up of tens to hundreds of millions of cells. All of them may be descended from one cell that was initially placed on the agar in the plate. And the cell is just divided from one cell into 2, into 4, into 8, into 16, into eventually a million cells before you got the colony. Cells usually grow by binary fission, and you can take a look on binary fission. We have a video for you to watch. And there's an incubator for growing media, supplying moisture. And the incubator usually warms up over room temperature. Here we can see bacteria colonies. You notice their border is distinct, clearly demarcated. They're smaller colonies than the fungal colonies. And you notice how they're fuzzy or hairy looking. And that's just that fungal colonies look hairy or fuzzy looking. Any questions on any of that? You do need to be able to distinguish colony characteristics, and this figure 5 shows you the different colony characteristics. The shape, what the shape of the colony looks like. The margin, that's what the edge of the colony looks like. So entire margin is one of like a circular colony where the margin is entire. Then the margin is undulate, lobate, curled, rhizoid, filamentous. The elevation of the colony. Usually you have to take the lid off the plate and look at the plate at an angle to determine the elevation of the colony. There can be flat colonies, a raised colony, a convex colony, a superconvex colony, which is called pulvinate, and then an umbunate colony that sort of looks like a mountain. I won't ask you to distinguish between convex and pulvinate. It's too difficult. And the same thing, I won't ask you to distinguish between rhizoid and filamentous, or lobate and curled. There's a size to the colony, a texture. The colony can be smooth or rough. And let's take a look here. Is that colony smooth or rough? This one here. Come on, somebody can state it.
Smooth.
Yeah, that's a smooth colony. Thank you. And is that an opaque colony, translucent, or transparent?
It's pretty opaque.
Yeah, if you can't see through it, it's opaque. Most of the colonies are opaque. Let me see another better example. Oh, here's a better example. So most of these colonies are opaque, but you notice on the edge here, maybe not in the center, but on the edge here, the colony is transparent. The same with that one, okay? There are some colonies that are transparent. Very, very few colonies are, excuse me, this is translucent. There's very few colonies that are transparent. Transparent colonies are hard to see. And so it probably wouldn't even show up very well here. Transparent meaning you can see through it. Translucent, you can see through it, sort of like muddy water, you can sort of see through it. And then opaque, you can't see through it. Most colonies are opaque, but some are translucent, and then very few are transparent. There's also a glistening colony. That's the appearance, whether it's shiny or dull. Is that colony shiny or dull?
It's kind of shiny.
Kind of shiny. I'm not sure if any of those are dull there, so let's go up to here. Not sure any of those are dull either. It might be hard to photograph a dull colony. Let's see. You can have colored colonies. Those are pigmented, and if there's any color to them, we call them pigmented. If they're not pigmented, we call them non-pigmented. And non-pigmented are cream, tans, or white, or off-white. And then with any color to it, yellow, white, red, we call it colored. Those aren't the best examples. Let's go up to this one. So these are kind of yellowish. That also yellowish, so those are colored. These are off-white. That one's white. So these are non-pigmented. But these are definitely, all of those are colored, pigmented. All right. You should know how to use this table or figure here. I'm not going to ask you questions without giving you this figure. So you don't have to memorize that undulate is looking like this and rhizoid is looking like that, because I'll give you the table. And like I said, I won't ask you really a nasty question like, is this colony undulate or lobate, because those are similar, or rhizoid or filamentous, because once again they're similar. But I will ask you to say something, is the colony circular or is it filamentous? Okay. And I'll give you this table to help you. Any questions about that? All right. You should know the terms abundance. That's simply the total number of colonies on an agar plate. And you just count all the colonies, regardless of how they look, how big or small they are. You count all of them. The diversity is the number of different colonies. And here there's a little bit of interpretation. You have to look at the colony characteristics and then decide, is different color, different shape, different texture, different other characteristics, allowing you to say that there's more than one colony diversity. That plate's too difficult to do, but let's go here. So all of these little yellow ones are probably one species. That one's probably two. This is obviously a third species. That's a different color, so that's a fourth species. That's a different color, so that's the fifth species. And then that's a different color, so there's at least 6 different species on this plate. So the diversity is at least 6, and you could go into different ones, like maybe that's a different colony there. Okay, but at least 6 different, diversity is at least 6. Maybe 7, and somebody could argue 8, okay? But something like that. And then there's about 30, I'm not going to count them there, but about 30 different, abundance is 30 colonies. Any question about any of that? The diversity, something like 6, at least 6. Moving down. So on the picture of this cat, how many, what's the abundance of cats here? 1, 2, 3, 4, 5, 6, 7 cats. The abundance is 7. What's the diversity?
2.
2, thank you. Yeah, there's cheetahs and lions. I think that's actually a question in your lab. So look over the terms. And then complete the laboratory exercises. You don't need to look at the references. One time somebody looked at all the references and there was a virus in one of the links. The instructors just put the reference there and nobody checked them. So write up your exercises to lab exercise 1. You can find that right here, worksheet lab 1, and it is due January 9th for 2.5 points. Remember to turn in worksheet lab 00 Friday, January 8th at 8 a.m. because I'm going to use that as attendance to see if you guys, if everyone's attending the class. And anyone who doesn't turn it in by that time will be dropped from the class. All right, any questions about any of that? If not, go ahead and work on the lab. I will be here at least until 7:30 to answer questions. Okay?
Hi, Mr. Volpe.
Yes.
For the lab exercise,
Uh-huh.
so one of the question it asks,